on the project that I did for my master's thesis at Humboldt State University with Mihai Temescu, that was published this past summer in IJPS. And when I talk about my master's thesis project to people, the most commonly asked question I get is, what is a zostrophil? Which is a fair question because Devonian paleobotany isn't exactly a common curriculum element. But to best understand what a zostrophil is and how to explain what they are, I need to discuss plant evolution on a broader scale. So with that, welcome to the last 425 million years or so of plant evolution, as fast as I can reasonably cover it using this cladogram that is representative of my artistic skills. And so I'm going to start with the plants that are alive today, the things that you're probably familiar with. The first being the non-vascular land plants, things like mosses, liverworts, and hornworts. While this area of the phylogeny is still kind of disputed, it's pretty commonly accepted that the common ancestor of vascular land plants resembled or was a member of one of these groups. And so the next group is the bulk of the critters you think of as plants, which are members of one of our major clades of vascular plants, the euphylophytes, which includes things like flowering plants, ferns, conifers. Here I have a monkey puzzle tree, as well as a number of other less recognizable plants. And they form a major clade here on our cladogram. And then finally, my personal favorite, which is the lycophytes. They were once a very diverse group, but today are represented by only a handful of taxa, things including spike mosses and club mosses. And they form our other major clade on this. And so now that I've given you your taxonomic bearings of these living plants to use as landmarks, I can illustrate the position of some of the relevant extinct groups more effectively that you will need to understand kind of what a zostrophil is. And so this grade that I've highlighted in yellow, falling between the moss or moss-like common ancestor of land plants and the split of our two major clades of vascular plants, is this group of plants called the rhiniophytes. Vascular tissues arose somewhere within this group. So some of the rhiniophytes and then the rest of the plants in this cladogram are vascular plants. And then the next group of plants is called the trimerophytes. They form a grade here at the base of the euphylophyte clade. And from these plants, most of the diversity that you know and love came from a trimerophyte ancestor. And then last but not least are the zostrophils, as promised. They are this group of Devonian plants here at the base of the lycophyte clade, and they form a grade similar to the trimerophytes. And so zostrophils inhabited the earth from the late Silurian to the middle Devonian, which was a relatively short span of time centered around 400 million years ago. And at one time had a worldwide distribution. Depending on whether you include some of the putative zostrophil taxa and gametophytes that have been tentatively assigned to the group, there are about 42 known genera. They're recognized by a couple key features, both morphologically and anatomically. And botanists tend to split morphology and anatomy into two things, where morphology refers to external or organism level general features of a plant, whereas the anatomy refers more exclusively to cellular and tissue level characters. And so externally, they're recognizable by these kidney-shaped or reniform bivalvate sporangia, where you can see one of the sporangial valves here, and they're laterally arranged on the axes. Ooh. Oh my goodness. There we go. And so anatomically, meaning if I make a cross section in the axis here of this plant, it's going to look something like this with your ground tissues. It looks like we don't have an epidermis, and then your vascular tissues, which is the xylem and phloem. This is not exactly the same because this is a zostrophyllum and this is a goslingia, but bear with me. It'll look something like this, where we can identify it as a zostrophyll because it has this round to oval shaped water conducting tissue called the xylem that matures centripetally and is composed of different sized water conducting cells called tracheids, where the smaller tracheids are on the far outside edge and termed protoxylem, and then we have larger cells towards the center, which we call metaxylem. And additionally, these zostrophills have a very specific type of wall thickening pattern where you get this ladder-like thickening with perforations in between the rungs of this ladder. And so to pivot a little, we classify fossils as either compression fossils or permineralization fossils, where compression fossils provide us with the best understanding of the morphology of a plant. And in these fossils, the external structure is preserved and we can best understand how the whole plant may have looked, things like branching patterns and sporangial arrangement. Whereas permineralizations, which are also much rarer than compressions, preserve that anatomical data that I was discussing, things like xylem characters and cortex histology. And so the relative rarity of permineralizations, as well as the quality of those we do have, hinders the inclusion of these anatomically preserved fossils in many studies. And historically, anatomy is a frequently undersampled facet of the data in phylogeny, potentially due to a combination of factors. The first being the scarcity of those permineralized specimens has lessened the inclusion of both plants known only from permineralizations, as well as utilizing anatomical characters to parse out relationships. However, the anatomy is an integral part of the structure of these plants and contributes to the characters that define them. So it's worth considering more in depth in phylogenetic studies. And additionally, a higher number of zostrophills are now known anatomically due to recent work compared to the body of data that was available in previous studies. And so with this in mind, we sampled zostrophills focusing on those whose anatomy has been documented. And doing this, we ended up with 19 zostrophill species from 17 genera, where we have 2 different species of Zostrophyllum and Saudonia. And then additionally, because the current understanding of tracheophyte phylogeny suggests that zostrophills, or at least some of them, are forming a paraphyletic group basal to the lycopsids, we thought it desirable methodologically to include a lycopsid representative in these analyses to polarize the characters. And so we included Singelia radicans, which is this early Devonian fossil lycopod that was described by my now advisor, Dr. Kelly Matsunaga, for her master's thesis when she was with Mihai. And so to investigate relationships among these taxa, we created a novel matrix of 40 characters, of which 26 describe vegetative morphology and anatomy. 12 of these are strictly anatomical, and 14 characters that describe the position and morphology of the sporangia. And using this data set, our main goals were to assess the influence of tree rooting, taxon sampling, and morphological versus anatomical characters on the resolution and stability of relationships, and then also to compare these phylogenetic results with phonetic or the more traditional numerical taxonomy methods in terms of what relationships we recover. And so I'm going to start by addressing the influence of tree rooting and taxon sampling on the resolution of these relationships. We experimented with 2 taxa when in rooting the phylogeny, the first being Xylophyton, which is a trimerophyte with well-known and characterized anatomy. And to remind you, the trimerophytes were that grade at the base of the euthylophyte clade. And so it's a stem group taxon of that clade's sister, the lycophytes, which makes it a good candidate for an outgroup. But trimerophytes are also very different from the lycophytes and zostrophils, which reduce the number of characters we could score for Psilophyton. And this other taxon, Rinalia, shares many characters with the zostrophils, but also has a number of rhiniopsid features. And rhiniopsids, as you recall, or at least some of them, are thought to be basal to that zostrophil trimerophyte split. And so then again, the anatomy of this plant isn't known. So that also reduced the number of characters that we could score for our analysis. So looking at some of these trees, we found that regardless of how we rooted it, there was no resolution in our strict consensus tree that included all of the in-group taxa. However, the majority rule consensus trees from these analyses with all the in-group taxa, and I've put one of them here, show some strong trends in our data set, namely being these two clades are consistently recovered in most of our trees. One of these clades includes our lycophyte representatives, Songelia, and 3 of the zostrophils. And then this other clade that contains the bulk of our zostrophil taxa, I've highlighted in green. It's interesting to note that in the analyses that we root with Psilophyton, where we still include Rinalia, more than half of these maximum parsimony trees recover Rinalia more closely to the bulk of our zostrophils than these other known zostrophils, Huya, Nathia, and Stolbergia up here. And so while we can't be entirely certain of Rinalia's placement without its anatomy, this gives us some indirect evidence that Rinalia may be a zostrophil. Because those analyses, the strict consensus trees that included all, strict parsimony trees, included all of our in-group taxa, lacked resolution, we experimented with removing taxa that had high amounts of missing data that's common in fossil data sets. And so these taxa are Stolbergia, which has 54% missing data, and Euthyrsophyton that had 44% missing data. And so when we excluded Stolbergia, we had a slight increase in resolution, but further exclusion of Euthyrsophyton resulted in the highest resolution of all taxon samplings. And so in this strict consensus tree that is rooted with Psilophyton and then excludes both Stolbergia and Euthyrsophyton, we are again getting those yellow and green clades that were recovered in the majority rule consensus tree that included all in-group taxa. And this consistent recovery of these two clades has interesting parallels with this non-phylogenetic hypothesis that was proposed by Nicholas and Banks, where they were looking at the origin of lycopsids, and they separated the zostrophils into a couple different groups based on the development of their reproductive axes. And the first way they parse them out is into groups based on whether these axes terminate in a strobilus or continue to grow after sporangia arise along the axis. And then further by the symmetry of these reproductive axes. And so we're going to focus on this non-terminate axis side of this flowchart. And this clade here that I highlighted in red for these purposes, but has been the green clade up until this point, aligns with this bilateral non-terminate zostrophil group here in the flowchart, and more specifically, the taxa that I've starred in blue from our analyses overlap with taxa explicitly included in this hypothesis. And furthermore, this clade here, which has been our yellow clade, which contains our lycopod representative that I've starred here, also contains this zostrophil discalus, which is included in this radially symmetric non-terminate pre-lycopod group and provides phylogenetic support for this hypothesis that the lycopod ancestor was likely a member of this non-terminate radially symmetric reproductive axis group. And so now we're still looking at analyses that are excluding our two taxa with the highest amounts of missing data. And these trees are strict consensus trees recovered when rooting with one of our outgroup taxa at exclusion of the other. And they have broadly similar but not identical topologies. We're still seeing those yellow and green clades that we've previously recovered. And this tree that we rooted with Renalia is also recovering this clade Huya and Nathia that I've highlighted in blue. But what I found most notable about these trees is that our two species of Zostrophyllum are recovered in different positions on in these analyses. And so in this Xylophyton rooted analysis, we have one species of Zostrophyllum parsing out with this yellow clade and the other as part of this green clade. And then in the Renalia rooted tree, both of them are part of this polytomy that is including our yellow and green clades. And so neither of these analyses are providing support for the monophyly of the genus Zostrophyllum, which suggests that the taxonomy of the genus may need to be reevaluated because it does contain most of the fossils described in the Zostrophyllophytina. And so to summarize this section, we found strong support for two major Zostrophyll clades that, excluding taxa with high amounts of missing data, improve the resolution of our trees, that our findings are generally supporting that Zostrophyll stem group of lycopods that was hypothesized by Nicholas and Banks. And some of these analyses support Zostrophyll affinities for Renalia, as well as Zostrophyllum not being supported as a clade, and we may need to reevaluate the genus. So now I'm going to pivot to talk about how morphological and anatomical characters contribute to phylogenetic resolution after I take a sip. So to do this, I broke our data up into two subsets of characters. One of them includes only characters that can be scored in compression fossils, which I've labeled here as morphology in quotes, which is specifically 26 characters. And then this other subset, which includes 30 characters that can be scored in permineralized fossils, which in quotes is anatomy, and I'll be referring to as anatomical characters. And so you may be wondering why these two numbers don't add up to the 40 characters that I was talking about at the beginning, which is our total number of characters. And that's because some of these characters can be scored in both compression fossils and permineralizations. And again, these analyses are rooted with Xylophyton and are excluding Stolbergia and Eutherosophyton. And so here are strict consensus trees that were produced using the morphological characters, which is here on the left, and then anatomical characters here on the right. And so it's interesting that the consensus topologies obtained using only morphological or only anatomical characters with the same taxon sampling are similar, but also have some significant differences. And so again, I've highlighted those two clades that we're consistently recovering. And I'll also point out that once again, we're having the two Zostrophyllum species parsing out in different places in these two trees. And so when we compare this morphology only and anatomy only tree with our all character tree with the same taxon sampling, we see again, there are differences in topology, and neither the morphology nor the anatomy tree is identically reflecting the results that we obtained from a complete data set. And so another key way to illustrate these differences is there are a couple of taxa that drastically change position, Discalus and Trichopterophyton, that I've indicated on these trees with the red and blue asterisks. And so while neither the anatomy nor the morphology trees have an identical topology to our all characters tree, both of them have better resolution than this all characters tree, which begs the question, which one is the best tree? And so because both morphology and anatomy are part of a species structure and arose as a result of its evolutionary history, analyses based on as many characters as possible provide the best supported and most accurate representation of relationships, biologically speaking, irrespective of the amount of resolution obtained. And so conversely, this also suggests that the traditional practice of employing only characters that are seen in these compression fossils or reproductive characters, which are thought to be more phylogenetically informative, may be producing trees that are not as accurately reflecting their true relationships. And so to summarize this section of our character sampling exploration, we found that trees obtained with different character sampling show differences, but maintain those same overall patterns. Subsampling characters improves the resolution of the tree. And 3, using as many characters as possible makes more sense biologically, even though it may lower phylogenetic resolution. And so finally, let's compare phylogenetic and phonetic methods in terms of the relationships that we recover. And so for those of you who may not be familiar, phonetic methods, kind of those traditional, numerical taxonomy methods, differ theoretically from phylogeny, namely because while in phylogeny, we're able to predict relationships and create a hypothesis as to how evolution occurs, numerical taxonomy can only assess and group taxa by their measure of similarity. And so these are 2 different dendrograms that we produced in a cluster analysis, UPGMA, where we used Jacquard's difference coefficient on our data set that excluded Xylophyton and Sengelia, because unlike in a phylogenetic analysis, you don't need to polarize your characters for a similarity comparison, pairwise similarity comparison. And so if we compare the similarity between taxa in analyses that are including or excluding those taxa with high amounts of missing data, it became very clear that these results, these clustering results, are highly sensitive to taxon sampling. And for example, these pairs of taxa are most similar to their partners in both dendrograms, these pairs highlighted in red, but the Goslingia cerulecalis pairing clusters with different taxon configurations in each of these analyses. On the other hand, we did see that Huya and Nathia are found in both dendrograms to be most similar to each other and most different to all the other Zostrophil taxa. And so this same relationship was seen in our phylogenetic analyses, where Huya and Nathia either form a clade or a grade outside of the clade that excludes the other Zostrophils. And so at least for these 2 taxa, the results of clustering and phylogenetics are concurrent. And however, when we compare the dendrogram and phylogeny with identical taxon sampling, we see that the clustering is not well reflecting the phylogenetic signal, as neither the green clade that we've been seeing nor the yellow clade are reflected in the similarity measures produced by this clustering analysis. And then the last thing that we noticed about these dendrograms is that Renalia is still consistently clustering with the bulk of the other Zostrophils. And here we're seeing at high levels of similarity in both analyses, unlike Huya and Nathia, which are known to be Zostrophils, giving us more support for Renalia having some sort of Zostrophil affinity. And then finally, we applied NMDS ordination, again, using Jacquard's difference coefficient to our data set, exclusive of Xylophyton and Sengelia. And the 3-dimensional configuration described 98% of the variation among these taxa. I color-coded the taxa according to their clade membership as they resolved in our phylogenetic analysis. We see this green clade here, this yellow clade that our lycopod was part of, Huya and Nathia in blue, and then the taxa coded in white are our 2 taxa with the highest amounts of missing data, Renalia, and then our 2 species of Zostrophyllum. And based on these 2 axes of similarity, we didn't see a strong phylogenetic signal in this ordination plot, with the exception of this grouping of most of the green clade taxa. And once again, we're seeing Renalia is closely associating with Zostrophil taxa, more so than some other known Zostrophils are, which provides some additional statistical support indirectly to suggest that Renalia may be a Zostrophil. And so to summarize, the phonetic portion of this, we found that these phonetic methods are not paralleling closely the phylogenetic results. However, clustering results are supporting our phylogenetic position of Huya and Nathia, and the ordination is again suggesting that Renalia may be a zostrophil. And so to conclude this talk, using 40 characters, we obtained a well-resolved phylogeny of 19 zostrophil species that preserve anatomy, with 2 main zostrophil clades supported, 1 giving rise to the lycopods, as suggested by that hypothesis Nicholas and Banks posed. Additionally, we found some evidence that partial sampling of characters may produce misleading phylogenetic results, and that there is an overall lack of congruence between the results of numerical taxonomy and phylogenetic relationships. And so to sum it up, I'd like to thank my funding sources from Humboldt State, the Jennings Family Botanical Society of America, as well as Allison, Nacho, and Santiago for their help with my phylogenetic analyses, and Eric Jules for his help with the ordination. I included my contact information over here if you have any questions, although who knows how long Twitter is going to be around at this point. And if you'd like to access this paper and read it, I have included a QR code here that links to the paper on my website. Thank you.
Okay, thank you everyone for being here. Thanks for the invitation. We're going to switch gears now. We're going to talk a little bit about phylogenomics. And there you go. So it's been clear for the last few decades that the advent of genome scale datasets has allowed us to tackle phylogenetic questions that once seemed unattainable. But today I'm not going to focus on discussing the robustness or statistical prowess of genome scale datasets. I'm going to focus on a different set of phenomena that have emerged during the genome scale era. And that is on the firstly, that phylogenomic data often harbor a mixture of signals for alternative trees. So here we have very famous contentious example of the position of horseshoe crabs. And while a vast majority of genes favor the controversial position of them nested within terrestrial chelicerates, there's also a large fraction of genes that favor the more traditional position of them as sister group to all the arachnids. And to some extent, phylogenomic inference is an exercise in weighting the multiple different signals contained within large data sets. But it's also true that not all genes are equally useful. And here we have 2 genes that I sampled from a phylogenomic data set of pseudoscorpions. And although, as you can see on the left, we have a gene that matches perfectly our understanding of the taxonomy of this group, the major subdivisions, and therefore probably has a well correspondence with biogeography and morphology, all these other sources of information which traditional taxonomy was built upon. While other gene trees fail to find any sort of meaningful grouping of these taxa and have a number of other problematic features, such as, for example, extreme heterogeneity in the rates of evolution between different terminals. And so these problems were already discovered in what can be thought of as the first phylogenomic study ever published, the 106 gene dataset of yeasts published in 2003 by Rokas. And they pointed out that single gene data sets generate multiple robustly supported alternative topologies. And they try to tease whether some of those genes can be deemed more useful in reconstructing the true relationships among these group of yeasts. And at that point, they say that they found no identifiable parameters that predict the performance of individual genes. Now, a reanalysis of this same data set that came out 3 years later by Geoffroy did in fact find predictors of the accuracy of gene trees. And they say that selecting only data that contain minimal non-phylogenetic signals takes full advantage of phylogenomics. And whether or not you agree with that, we can argue that phylogenomic subsampling was born and that which we can define as building trees using only some of the thousands of genes that are available today. And there's various reasons why someone would like to do this. We've covered reason number one, that's to discard loci that are deemed misleading. So some people have argued that the analyses that come out of entire phylogenomic data sets are probably not accurate for various reasons. And so discarding part of the data might give us a better result. Alternatively, there's another reason, and that's just computational limitations. A number of inference types of phylogenetic inference that can be done today are still very far away from being computationally tractable with thousands of genes. And so when one goes to the phylogenomic subsampling literature and see what people have done in the past, the vast majority of studies have focused on subsampling data sets based on a single property. And there's generally sort of two flavors to this approach. Some people have sought to minimize evidence of systematic biases in the data. Other people have sought to maximize proxies of phylogenetic signal. And here we have two empirical distributions of a data set that I built for sea urchins. On the top, a metric considered a source of systematic bias, the average pairwise patristic distance of the gene trees. The larger that is, the more likely that a gene tree will suffer from long branch attraction artifacts. So it's something that ideally we would like to minimize. And on the bottom, we have a proxy for phylogenetic signal, which is the average bootstrap support of the gene tree. And so these two different approaches, what they look like, empirically speaking, is something like this. We would apply a given threshold and retain for example, on the top, a given fraction of loci that are least affected by biases, colored in red, or on the bottom, a given fraction of loci with the highest evidence of phylogenetic signal. Now, what no one had looked at up until that point is how different these two approaches are. Maybe, potentially, they're just sampling the same sets of highly reliable genes. Maybe they're favoring completely different types of genes. No one had really looked at this before. But if in fact they are different from each other, they're favoring different types of genes, then is either one of them preferable to the other? And to answer these questions, we're going to look at the same data. Here I'm just showing the bivariate distribution of genes on the same two axes. On the x-axis, we have our source of systematic bias, and on the y-axis, our proxy for phylogenetic signal. And each gene here is represented by a dot. And when we're subsampling the data set, we're essentially, again, just applying these thresholds and selecting just a fraction of the total data. And you can see right away that these two approaches are extremely different from each other. They have a minimum degree of overlap. Even when we're sampling, I think 30% of the data for each one of them, they only agreeing on 4% of that 30%. So a minimal agreement on the genes that should be selected for downstream analyses, and therefore probably a large difference in our final results, whether we choose one or the other strategy. So given this result, can either one of them be defended as being more preferable than the other? And to answer this, we're going to focus on the centroids of the distributions. Here in gray, we have the centroid of the entire distribution, and in red, we're showing the centroid of the distribution after applying this threshold and selecting only the genes that have the minimal amount of average pairwise patristic distances. So minimal signal of contributing biases to the analysis. And when we apply this threshold, we're not only losing biases, which is our intended effect, but we're also having the unintended effect of losing signal. And conversely, when we try to maximize signal, we're also increasing the amount of biases. So strictly speaking, neither one of these is preferable. Neither is doing what we want it to do, which is select genes with high phylogenetic signal, while at the same time having low evidence of systematic biases. And the reason why this doesn't happen is because both of these properties are positively correlated with each other, such that we cannot subsample based on any one of them individually without either increasing both or decreasing both at the same time. And so this pattern of correlation is sort of confounding the analysis by introducing effects on properties that are not being considered if we're subsampling just one property at a time. to show you another example of this, it's become very common within people that are trying to estimate divergence times to use genes that have the most clock-like evolution. And so a common way of selecting for clock-like genes is minimizing the root-to-tip variance of the gene tree. And while this has been validated as a way of attaining accurate divergence times in simulation frameworks, when we deal with the vast ranges of evolutionary rates that are common in empirical data sets, minimizing root-to-tip variance is the same thing as minimizing the rate of evolution. And the reason for this is clear. The most clock-like gene is a gene that does not evolve at all. And so when we're trying to find clock-like genes in empirical data sets, we're actually just selecting for entirely uninformative genes that look like this, genes that have almost no phylogenetic information within them because they're pretty much not evolving at all. So another approach that I came up with is based on the simultaneous use of multiple properties. We've shown that using just a single property one at a time is confounded by the correlation and the effects in non-targeted properties that correlation generates. So maybe a way to work around this is to explicitly use the pattern of correlation among multiple properties. Maybe that would give us a way to discover better access within within what is essentially a multidimensional problem with which along which we can subsample genes. And so we perform a PCA analysis on 7 gene properties, and these were selected because they're a number of properties considered to be conducive to systematic biases. There's some that are considered proxies or phylogenetic signal, and there's one that's just a metric of the overall information contents of the genes. And we were very surprised at first to find out that the first single PC dimension that's found accounts for 50%, roughly 50% of the total variance within this problem. And as you can see, when the scores along PC1 are correlated with an independent estimate of the rate of evolution of the genes, then we see this almost linear correspondence between the two, such that we can say that PC1 broadly is recapitulating the rate of evolution of the different genes. And this is the major aspect of variation among all of these 7 properties. And when we look at the loadings of these gene properties onto PC1, we see that as we increase the scores along PC1, as we evolve faster and faster, then genes simply attain higher levels of information, higher levels of phylogenetic signal, but also higher levels of systematic biases. And this is also an expected result. Genes that do not evolve at all or evolve very slowly probably don't have any of these. And as rate of evolution increases, then all of these properties increase sort of concomitantly. Now, what's really interesting is that the second major PC axis, which I explained about a fifth of total variance, is roughly uncorrelated with the rate of evolution. There's some sort of peak at intermediate rates of evolution, and that's going to become important later on. But when we look at the loadings, we see that selecting based on PC2, or the scores of genes along PC2, enriches the data in genes that are highly informative, have a high level of phylogenetic signal, but at the same time show evidence of reduced systematic biases. And that's exactly what we wanted a subsampling method to do. And that's why we term the second PC axis a phylogenetic usefulness axis. And we're very happy with this result. We just, for this study, we subsample a small number of genes and run our phylogeny with that. But it also left us with a number of questions. Namely, are these patterns of correlation common to all phylogenetic data sets, or are they just something that we happen to discover in this particular matrix that we were working with? Or another way of saying that is, can we estimate evolutionary rate and phylogenetic usefulness of the genes simply from a combination of gene properties and their correlation coefficients? And furthermore, how does this multivariate method of subsampling that I just introduced behave in comparison to other options that have been used in the literature before? And so to tackle these questions, we gathered a sample of 18 phylogenomic datasets from the literature, which span different clades, different number of loci from about 1,000 to 4,500. wide range of taxa, and also a huge disparity in the age of the phylogenetic questions. And with this data, we sought to answer first, are these the same two underlying axes present across all data sets? And we do in fact find the same two axes, one along which all 7 properties increase, have positive loadings. Here I'm showing you the distribution of the loadings of all of these 7 gene properties onto PC1 along the 18 data sets. And you can see that on average, although there's an axis, there's a first PC dimension along which all increase concomitantly. And there's a second axis that we referred to before as our usefulness axis that has this distinct pattern of increasing phylogenetic signal while at the same time decreasing sources of systematic bias. So the answer to this question is yes, we can estimate evolutionary rates and phylogenetic usefulness of the genes based on just these 7 gene properties. But we haven't yet tested whether the trees that are found using this small subsample dataset obtained with our subsampling method are actually conducive to generating accurate phylogenetic trees. And so to kind of test this, we started with our full dataset and we performed a tree inference using the entire data. And we also performed 22 different subsampling approaches. 5 of these were just replicates of selecting genes at random, and 17 other matrices were built using targeted subsampling approaches, things like recovering the slowest evolving genes, the ones with the highest average bootstrap support, et cetera, including also the multivariate method that I just introduced. And these matrices were relatively small compared to the full data. So we then performed tree inference on each one of these 22 data sets, and that gave us a distribution of trees, which we compared to the tree obtained with the full data set using Robinson false distances as some sort of measure of topological similarity. So this gave us a sense of how far these topologies are from the tree inferred using the full data. And what we did is, first of all, average all of the 5 replicates that we obtained using randomly sampled genes, so into just one sort of average topological similarity that can be expected from small data sets. And then we used this metric of Robinson false distances to sort different targeted subsampling approaches from the ones that recover the topology most similar to the entire data set to the one that recovers the one most dissimilar. And the problem here is that we would be tempted to use this as a proxy for the accuracy. But the problem is that we don't really know the true underlying tree. This tree obtained using the full data set is not necessarily correct. And in fact, a large driver of why we're subsampling in the first place is maybe because we don't even trust the tree that we obtain with the full data set. But it's also true that these small subsample data sets, the tree that they favor, should broadly recapitulate most of the nodes in the tree obtained for the full data set, while being allowed to differ in a number of ways. So broadly speaking, shorter distances are good, but the shortest distance is not necessarily the best. And so to account for this, what we did is not consider necessarily the rankings that we obtain, but simply split these different strategies into those that perform better at sampling genes at random and those that perform worse at sampling genes at random. And this is just a fact that we're favoring topological similarity to some extent, but not being too strict on it, but also asking from a method to perform better than just sampling genes at random, because otherwise that's probably what we should be doing. And there's no point in actually devising any sort of technique. What I'm going to show you is not just the numbers obtained under one data set, the rankings obtained under one data set, but the average ranking across all 18 data sets that were used. And so this is what it looks like. Here, that gray dotted line shows the average performance of random loci across all different data sets. And what emerges right away is that there's very few subsampling methods that perform better than random sampling. Luckily, the one that I just introduced, PC2, selecting the highest scoring genes along PC2, or the highest of the genes with the highest phylogenetic usefulness as we defined it, is one of those 5 approaches that perform better than just getting genes at random. But maybe more interestingly than this is that the most common approaches that you can find in the literature, sampling based on rates, which is shown here in yellow, and sampling based on sources of bias, minimizing them, fare worse than random sampling. And this is a very intriguing result, and we would like to know, why do these commonly used approaches fail to produce accurate trees when there's simulation studies and phylogenetic theory that suggests that these are valid methods. And I'm not going to touch much upon why minimizing bias is a bad strategy. I already introduced an example that showed that in general, when we're dealing with empirical data sets, minimizing biases such as saturation or compositional heterogeneity is more or less the same as minimizing rate. The least saturated genes in an empirical data set, the least compositionally heterogeneous, the ones with the least rotative variance or the most clock likeness are all the ones that are evolving really slow or not evolving at all. So the reason why this fails is because they're generally just getting at genes that are uninformative. But this still leaves us with the question of, why is evolutionary rate such a poor proxy for signal, when theory predicts that there's an optimal rate of evolution that maximizes the phylogenetic usefulness of genes given a phylogenetic question of a given age, for example. This has been shown both theoretically and with simulation methods. And we do recover a similar pattern in our data set. So here is just one of those data sets, the Lepidopteran phylogenetic data set. And you can see that there is a peak of phylogenetic usefulness at a given rate of evolution. But there's also an extremely wide spread of usefulness for that optimal rate. So what we're going to do is just plot this in a different way. We're going to just average out the usefulness for 10 different bins of genes evolving at similar rates of evolution. So we partition the rate of evolution into 10 different bins, and for each one of them, we compute the mean usefulness, but also the variance in usefulness of the genes evolving under that rate category. And what you can see is a perfect linear correlation. So the genes that are evolving at the rate that maximizes usefulness also show extreme variance in their usefulness, meaning that on average, they're good, but a lot of them are really, really bad. And so I think that evolutionary rate is not such a good proxy for signal, because it's only part of the equation. Useful genes do evolve at an optimal rate, but not all genes evolving at that optimal rate are useful. So the converse is not true. And that is why I think we can explain why evolutionary rates in general, even if we zoom in into what's thought to be or estimated to be the optimal rate of evolution for a given phylogenetic question, we still do not recover accurate trees with some subnode data sets. And so to end my talk, I'm going to go back to this paper that was published more than 15 years ago. And they say that we therefore suggest putting the emphasis on the development and refinement of objective methods aimed at detecting and removing the part of the data containing a high level of non-phylogenetic signal. And in the last 15 years, there's been minimal to no progress into developing these objective methods. Univariate subsampling methods that are the ones used by most people still today are to some extent arbitrary. It's hard to justify why one would go, for example, with maximizing signal versus minimizing biases. They're confounded by correlations that introduce effects across properties that are not directly targeted. So by not accounting for the multivariate nature of the problem, we're unaware that we're maybe introducing problems on other properties that are not the ones that are targeted. But they're also largely ineffective in supporting accurate trees. Minimizing biases, like we showed, selects genes that have almost no phylogenetic content. And even genes that evolve at optimal rates can also be uninformative due to other reasons. On the opposite, multivariate subsampling methods, such as the one that I introduced, reduce the arbitrariness by using multiple properties simultaneously. They also exploit the correlations across gene properties to discover an axis of usefulness. They have been shown to be effective in supporting accurate trees, even using small data sets. But I think more importantly, they're entirely data-driven. So the different variables that we're introducing to the method are going to be weighted based on how important they actually were during the evolutionary history of a given clade. So if there's been a lot of compositional heterogeneity, then that variable will load strongly onto a phylogenetic usefulness axis. If there wasn't, then it won't load. And that's something that's determined dataset by dataset without any input from the researcher. And so the politicians underlying this talk are those two mentioned at the top. And the software is currently implemented as an R script that I've named Gene Sorter. It's available on my GitHub repository, and I'm really happy to see that it's being used quite widely in the last year, not only by myself to work on echinoderms, but by people working on chelicerate and mollusks and even angiosperms. And with that, thank you all. And if there's time, we can go to questions.